Today we are going to finish the story that I've been teasing you about since last week. We're going to finally put everything together and make a protein from this mRNA using the mRNA, the tRNA, and the rRNA. So we'll talk about all three of these again. I know we've talked about them before, but we're going to go into more detail. Now we're going to tell you about how the mRNA is going to code for those amino acids. We're going to use those bases in sets of 3 that we call codons to code for specific amino acids. We'll have something called a start codon that will do initiation of that translation. That might seem counterintuitive to you. For me personally, I thought it made sense that you have to have a start site to do transcription. But if you have a piece of RNA, you've already edited it and you've sent it out, I just kind of thought it would read the whole thing. Turns out that's not how it works. You do have to have a start site, primarily because you have codons. So because you're reading them in 3 pairs, you have to make sure you start at the right spot, or you will be off for everyone you're reading. You'll also have a stop codon that's going to stop the translation process, and we'll talk about the different codes for those. For tRNA, we're going to actually show you that structure with those 3 hairpin turns on them, those hairpin loops, and we're going to show you where the anti-codon is going to be. That's a complementary base set to the codons on the mRNA, meaning that these tRNA now can come in and bind to the correct codons on the mRNA and bring in the appropriate amino acid to match. In order to get the amino acid onto the tRNA, we're going to use another enzyme called an amino acyl transferase. Then we're going to get into the ribosomes. So before we've been drawing these as just like these blobs. Now we're going to show them as 2 blobs, because now they're 2 subunits. So this is going to be a small ribosomal subunit, which is responsible for binding the mRNA and reading it to find that start codon. And then it will bring in the large ribosomal subunit. And this one is where the tRNA can actually bind and where it will drop off that amino acid that it was carrying. And the large ribosomal subunit will catalyze peptide bonds between each of those amino acids to grow the peptide chain. Then we're going to look at the whole story together. And we'll start with the simpler of these, which is just cytoplasmic, free ribosomes. We're going to put all of those together and see how we're going to make the protein and release it. But we know that we can't just do that if it's a transmembrane lysosomal or secreted. It's not going to be in the cytoplasm. So we'll talk about that signal to get it to the endoplasmic reticulum. That's a signal peptide. That signal peptide will be recognized by a signal recognition particle, moves it to the ER, where it will come in contact with a translocon. This is a transmembrane protein that's just a pore in the ER membrane. You're going to then continue synthesis of your protein through the translocon. And if you have a transmembrane protein, you'll have stop and start transferase that will get caught in the membrane so that you have those membrane spanning portions. It's tricky. Not necessarily because it's complex, although there is a lot of steps. It's tricky because it's really hard to draw, especially for me. And so it's just something that we're going to have to try to go over a few times. We'll show you some pictures. And I would highly recommend that you go watch the video under Tools for Success. It makes it so much easier to see it visually happening. If we have time, I'll try to show you that video, but it seems unlikely. So that'll be, well, I don't want to say homework for the weekend, but I think we all need our weekend. Certainly could, though. Wouldn't hurt. All right, let's start with RNA, and specifically mRNA. So we know that mRNA is the blueprint for we used 4 bases to make this. Those were going to be the guanine, cytosine, and uracil. And now we have to use these to code for the amino acids. So already you could see a problem, because with only 4 bases, if we used each one to code for an amino acid, we would be able to code for a total of 4 amino acids. We have 20. Even if we started trying to do those in pairs of 2, we'd get 16 total. So we have to do them in sets of 3. I'm just going to draw these in real quick. Okay, so this is our mRNA strand. I'll remind you too that we made this off of DNA and that we always read DNA 3' to 5'. We make a new strand of DNA 5' to 3', and we make RNA 5' to 3'. We're also going to read RNA 5' to 3'. So we'll say that this is our 5' end, and this is our 3' end. These codons are 3 base sets specific to an amino acid. All right, so you can imagine if you use 3 base sets, you've got 4 bases to choose from, you're going to have 64 possibilities. We only need 20. That means that there's some redundancy in there. You do not have to memorize any of them. Well, I'm sorry, you have to memorize one. You got really excited, but it's just one. We'll ask you to memorize the start codon. Other than that, you do not have to know the codons or the appropriate amino acids for them. Just understand that each codon has a specific amino acid and that you'll actually have multiple codons that can code for the same amino acid. So with your start codon, to start initiation, you're going to have AUG. That I want you to know. And I want you to know that this codes for methionine, which you can abbreviate as MET. All right, your stop codon terminates translation. It does not code for an amino acid. I'm going to use AA as an abbreviation for an amino acid. So if we're looking at this mRNA strand that we've created, we can see that as you start going through here, you hit an AUG. This is your start codon. This is also going to set with something called the open reading frame. So again, if we're going to sit here and read these in 3 pairs, 3 bases, if we start at AUG, then this becomes our next codon. All the way down. I actually counted right, yay. If you don't set this open reading frame correctly, if something happens and maybe you're slipping and you come back to this A instead, you're going to read AAU, GGA, GUG. Those are going to code for completely different amino acids. So your protein could be entirely different and non-functional. So you need to keep your reading frame by setting it properly at your start codon. All right. Again, you guys don't have to know this, but just so I can use this as an example later. GAG is glutamate, and UG is tryptophan. ACC is we'll just call that amino acid 4 because I forgot. And then this is your stop codon. Okay, so that's your mRNA coding for the amino acids that are going to go into the protein. Questions on mRNA? Yes?
So we only just have to know the start codon, none of the stop codon?
You only have to know the start codon, not the stop codons. If you want to know these, because you'll probably need them for another class, there's an easy way to remember them. You are annoying. You go away. You are gone. So this first one, UAA, you are annoying. You go away. You are gone. That's kind of rude, so if anyone comes up with a nicer version, let me know. I'll use that as an example next time. All right. Yeah.
What open reading frame is again?
So open reading frame, that's reading frame means where you start your reading. And once you start at one place, you have to read 3 at a time. So if you start reading at the wrong place, you're going to read the wrong 3 and set for different amino acids. Okay. Let's talk about tRNA. So I said that tRNA had those 3 hairpin loops to it. Oh, I want to do this in green. Yeah, I should do this in green. That's a vaguely accurate representation of a tRNA, 2-dimensional. Okay. I'm not going to sit there and draw tRNAs out like this every time. So in the future, I will draw them like, that's my tRNA. But you can actually see loops here. And of these, the only one that we care about in this class is this one at the bottom. Because this is where you're going to have your anticodon. Each tRNA is specific. It has a 3D shape and an anticodon. That means it can only pick up certain amino acids, whatever amino acid it codes for. If we were trying to find the tRNA that could match our start codon with an AUG, we would need the anticodon, which is UAC. And if we had that, then this guy could have a methionine attached at the stem. So that's where you attach the amino acid on these. Mm-hmm.
So could you explain again what the anticodon is?
So this anticodon is going to be complementary. I don't like complementary bases to the codon. The idea here is now we can take this tRNA and it can bind to the start codon. Okay, so the tRNA will find the appropriate codon that it can match with its anticodon, bind, and that's a signal to release its amino acid so that the ribosome can take those and peptide bond them together to create that protein. Yes?
Does it always bind to the start codon, or is it
So it's always going to bind to what it has an anticodon for. This example is an anticodon to a start codon, but you will have tRNAs for all of the possible codons. So we could have another tRNA that's going to have CUC. That was a bad example. Where is, there we go. That looks a little too G-ish. That's a C. And since that is an anticodon to this GAG, which codes for glutamine, or glutamate, not green, blue, then this amino acid would be glutamate. Okay. So the anticodon will always match a particular codon, and it will carry the correct amino acid for that. Yes?
So when it binds, what does it do after that? How does it distinguish? Like, does it just take that codon?
Ask that again.
Or is it connected to the RNA?
So when the tRNA binds to the mRNA, what was the question?
When the tRNA binds to the mRNA, is it then connected, or does it leave the mRNA with the codon on it?
So what happens when this binds? And we'll walk through the whole story, but just so you know here, this, just like any other nucleic acid bases, can hydrogen bond together. So when the tRNA comes in contact with a matching complementary set, you'll create hydrogen bonds between them that will hold it there temporarily, and then it can drop off the amino acid that it's carrying. It will then release and move on, and it has to pick up another amino after that. Okay.
So in the picture that you have on the board, is it already bound? Like, is the tRNA and the mRNA bound to each other?
In this picture, I'm not showing tRNA and mRNA bound. This is just the tRNA right here and right here.
Is the amino acids, the blue at the top, is that from the mRNA?
So this amino acid is going to get picked up in the cytoplasm and put on the correct tRNA, and we'll talk about how. And then this can then move to the ribosome to drop off the amino acids. Okay.
The only time to get methionine is when you have a start codon, and then every other one after that will have like a different amino acid.
So the question is, we'll have the start codon with methionine, always going to be methionine, and after that we'll have different amino acids. Sometimes you can have multiples. You can have, several lysine, well, probably not, but you can have several amino acids in a row. It depends on the code within the mRNA, but each one will be a specific amino acid. Okay. All right, so we can imagine now, and we'll imagine, I'm going to draw it out in a minute, but for now, imagine that this is bound to the mRNA. It released its amino acid. So glutamate's over here, now part of this structure, and we now have this empty tRNA. In order for this to still work, we need to put another amino acid on it. And so we have a cytoplasmic protein called an amino acid. Okay, name tells you it's going to transfer an amino acid. Again, these are very specific to the tRNAs. So all of this is happening in the cytoplasm. These are cytoplasmic proteins. Yep. So if this amino acyl transferase was able to bind to this particular tRNA with an anticodon of CUC, that means that this amino acyl transferase can catalyze glutamate being put back on to this tRNA. So now you've recharged this tRNA, and it can go back to the ribosome, see if it can find a codon to match again, which would be GAG, and if it does, it'll bind and drop off a glutamate again. And then it just keeps cycling through that. Yeah?
Wait, so why does it drop the glutamate just to grab another one? Is it dropping the glutamate into a ribosome?
Yes. So it's going to drop that glutamate into the ribosome so that it can make that glutamate part of a growing protein. And then it has to pick another one up to be able to bring it to the ribosome to drop it off. All right. Yeah. I'm sorry. So you'll have amino acids in your cytoplasm, and you'll have things like the amino acid transferase that will catalyze putting that onto the correct tRNA that it recognizes by 3D structure. Yes?
Will like the tRNAs change what their anticodons are, or is that just set for that specific tRNA?
So do the tRNAs ever change their anticodons? No, this is the base in that tRNA. This is how this tRNA was made. So once this tRNA is made, whatever anticodon it has, that's what it's set for. It will always carry the same thing. Yeah. Yes?
Does the tRNA always have that amino acid transferase on it?
Does the tRNA always have this amino acid transferase on it? No. This is going to come in just to replace an amino acid if it's empty. And once it puts it on, the amino acid transferase drops off, and the tRNA is able to go about shuttling the amino acids to ribosomes again.
So are there 20 tRNAs specific for each of the 20 amino acids?
I'm going to say yes, but it's a bit of a fib. For you guys, yes, there is a tRNA for every amino acid, for every codon.
Right, yeah.
Yes.
Does the body, or does the cell have a way to correct or get rid of tRNAs that have a mutation in that anticodon place so they bind to
So if your tRNA happened to have a mutation, say you had a tRNA that was supposed to be UAC and it was supposed to have methionine on it, but you mutate this just down here, let's say, not, because it's the entire 3D structure that affects whether it will bind to the appropriate amino acid transferase. Just a mutation down here would probably mean it would try to bind to an amino acid transferase for a MET, but it won't recognize the anticodon, and so this would just be non-functional, and it would get destroyed. Yeah. All right, let's kind of put this all together and walk through the synthesis. So we're going to start with, oh shoot, I forgot to tell you about the small and large ribosome. We'll just draw them here. Okay, so this is going to be translation now. So as a reminder, you had, I'll just kind of draw this. This is our nucleus. What is with these? Okay, we sent RNA out through a nuclear pore after it was edited. So now we're in the cytoplasm. First thing that's going to happen with this mRNA, it's going to get picked up by the small ribosomal subunit, which I'll just draw as a small ribosomal subunit. It's going to grab this mRNA strand. It's going to read it. We'll say it was a strand up there. So it finds the AUG, that start codon. That's going to recruit the large ribosomal subunit. Okay, so our next ones were GAG, and then we had UG, and then ACC, which I'll have to look up, and then our stop codon. So now we have our large ribosomal subunit. This is going to be the binding site for those tRNAs. So you'll have a tRNA that can come in, and it will have those complementary bases. So this for A would be a U, U is an A, G is a C. That was our complementary start, our anticodon for the start codon. And this then would be carrying methionine. We could have another ribosome now come in to this next site. It would have complementary CUC, and this is going to carry glutamate. Again, these codons and the amino acids are not important. I'm just using them as examples. Yes?
So are there going to be 2 codons in every large subunit?
How many codons can you fit within the large ribosomal subunit? There's actually 3. There are 3 tRNA sites in these. We don't get into that detail here, but yeah. Go ahead.
The first tRNA is carrying UAC, right?
The anticodon here is UAC, correct. So the anticodon for the second tRNA here is CUC. And again, those are going to be able to hydrogen bond with the codon, holds it in place. Now you've got these amino acids here. And the large ribosomal subunit can catalyze a peptide bond between them. Yes?
So how is this all oriented? Like the mRNA only has the on one side, so how does the small site read it and then the large site?
So the question is, how is this oriented? If we have a strand of RNA, we know that one side of it is going to be the backbone and that you will have those bases coming out of the other side. So if your small ribosomal subunit is reading one side, how is the RNA, the large ribosomal subunit, actually able to have this position for the tRNA to come and bind it? That's a great question. I don't have a great answer for it, except that this is a 3D structure. And so there is more ability, it's not always just going to be sticking straight up for the small ribosome. It may be more flat so that they can both. Yeah. Yes.
So the large ribosomal subunit catalyzes the peptide bond between the amino acids of the tRNA.
So the large ribosomal subunit now is responsible for catalyzing that peptide bond between those amino acids that are coming in. And as it does this, these tRNAs will release those, and these can leave. There'll be these empty tRNAs now, but they will still have that anticodon that will allow them to match perfectly with an amino acyl transferase and pick up another protein. They can then come back and wait and see, can they find another codon to match with? In this case, in this example, this wouldn't match again. We don't have another AUG. We don't have another GAG, so this one's pretty much done for this. But there's plenty of time. So these could maybe move on to another ribosome that's doing and find a matching codon. How does the large ribosomal subunit attach to the small ribosomal subunit? I didn't say. I'm not entirely sure, and you guys certainly don't need to know. So the small ribosomal subunit does recruit in the large ribosomal subunit only when it finds a start codon. So I'm not sure what the factors are exactly that makes the small and, yeah.
When does it have the tRNA leave the large ribosomal subunit?
Why does it have it leave? When? Okay, so when these bonds are formed between amino acids that are being held by the tRNA, this is going to release the tRNA, okay? It's going to be a signal that this doesn't need anymore. It gets kicked out of the site. This entire ribosome will shift. It basically walks along the mRNA, always in the 5 to 3 prime direction. So it's moving that way, 5 to 3 prime. And then it will read the next set. That will allow more tRNA to come in and find and bind to a codon that it's complementary to. It will have its protein on it. Trying to draw this in would be crazy, but I'm just going to pretend that we had this tRNA here. It had tryptophan. You would catalyze the bond between those. You've got a protein of methionine, glutamate, tryptophan, and you just keep moving down and adding additional proteins as they're matched, create a peptide bond between them, and then you release that protein. Yes?
So this process is making the primary protein structure that we talked about a few weeks ago.
Yes, this process is making the primary structure of those proteins. It's just pop beads, putting those amino acids together in a certain order that will then determine the secondary and tertiary. Yes. Yes, ma'am.
So are the tRNAs just like in the cytoplasm and then they're like entering the large ribosomal subunit? Like how does it work as it moves along?
So the tRNAs are in the cytoplasm, and as is the whole ribosome actually. But yes, the tRNAs can move in, try to find a codon that it matches. If it does, it will settle into the particular site in that ribosome, the large ribosomal subunit, and then be able to bind to the codon.
If it doesn't match, does it
Just moves on.
leave?
Yep. Yeah, it won't be able to sit in the site that's in the large ribosomal subunit.
Are the peptide bonds formed like when it enters the ribosomal subunit, but then the ribosomal subunit also breaks the peptide bonds?
So we won't be breaking peptide bonds here. Okay, I think I see where you're going. This is not going to create a peptide bond unless you have the tRNAs in the appropriate locations. We don't talk about those 3 different locations that they sit in, but basically you'll have 3 set at a time. You'll connect the 2, it moves everything. Then you connect the next 2, it moves everything. So that's creating peptide bonds between amino acids. If you're referring to breaking the bonds between the tRNA and the actual amino acid it's holding, that's a covalent bond, it is, but it's not a peptide bond. And that's powered again by the large ribosomal subunit.
So they're like attached, so the proteins attached to the site and they keep those peptide bonds together, or the amino acids, I mean?
So the protein is kind of going to be like free. We're drawing it here, but if you were to draw it here, it would basically make this protein, and as it moves along, it would just keep pushing it out, and you would just get your protein until it's done. Okay, so that doesn't stay in the ribosome, that actually gets pushed out. Does that make sense? Okay. We'll do this a few times, especially when we're talking about training. And like I said, definitely give that video a try. It's really nice to see it in 3D visual form. Yes?
So I guess like as the tRNAs are moving along and the peptide bonds are forming between amino acids, that methionine is the only amino acid that stays connected to that first tRNA as it moves along.
So none of the amino acids are going to stay connected to their tRNA. All of them were going to come off.
But I guess I mean like is methionine the last amino acid to come off of that tRNA?
No, methionine will be the first amino acid to come off because as this makes this bond, this amino acid will fall off. I shouldn't say fall off, it's a covalent bond, it has to be broken off, but it will come off. And then when this moves, this is bound to the next one with the peptide bond. That will cause the release of this one.
Oh, okay.
Okay, so you just keep releasing them as you move along, and it makes the...
Be the first one that's released.
Yes, exactly. Methionine would always be your first amino acid in your protein. Yeah. Okay. you'll just keep moving along doing this, adding more, making this bond, dropping it off the tRNA until you get to UGA. UGA is one of our stop codons, and there is no amino acid that matches this. Okay, so no amino acid gets brought in here, and that's a signal for termination. When this signal for termination occurs, what did I just stop codon as? Green. Your stop codon will cause the disassembly of the ribosome and release of that protein. So that means that once you hit the stop codon, the small and large ribosomal subunits will separate again. The mRNA that was sandwiched in between gets released, and the protein will finally fall through, and you'll have a full cytoplasmic protein. So maybe you just made a new amino acid. Okay. Maybe you made a tubulin to make microtubules with. But if you wanted to make something like a transmembrane protein, you can't do it here. This is just for your. So it gets. If we're doing transmembrane, lysosomal, or secreted. The process is actually going to start exactly the same way. You will push mRNA out of your nuclear pore into the cytoplasm. So you'll have a strand of mRNA. Ribosomal subunit will come in and bind to it. The AUG. It will bring in the large ribosomal subunit. And it can start making the protein. I won't show the tRNAs again, but you'll have the tRNAs come in. They'll start releasing the amino acids. Those are bonded together through the peptide bonds, and you start releasing a protein. So here's your start of your protein. But if this is meant to be transmembrane, lysosomal, or secreted, the start of this protein is going to have a signal peptide. That's better. And that signal peptide is going to be approximately 17 to 30 hydrophobic amino acids. So the first 17 to 30 amino acids that you put together for this protein will be hydrophobic, and you're making them in the cytoplasm. They're not happy about this. They don't like being here. Fortunately, the signal recognition particle can come to the rescue. Sorry if that looks an awful lot like my amino acid transferase, but this is a signal recognition particle, meaning that it recognizes the signal peptide. This is a cytoplasmic protein. It's going to have primarily hydrophobic R groups outside, so it's perfectly happy in the cytoplasm. but it's going to have a binding pocket inside that's hydrophobic. You can think of it kind of like a hot dog bun. If you open it up, it can actually close around that signal peptide and hide its hydrophobic interior and the signal peptide itself from that cytoplasm. Okay. Repeat that? Yes. So the signal recognition particle can be like a hot dog and picks up the signal peptide, so that the hydrophobic portion of the signal peptide and the hydrophobic portion of the interior of the signal recognition particle are now hidden inside this protein and away from the cytoplasm. Okay? This is like a tugboat. Okay? This is going to carry the entire complex to the endoplasmic reticulum. I should have done that up there. So, here's our endoplasmic reticulum. This side is meant to be the lumen of the ER. So that's the inside of the endoplasmic reticulum. This is the cytoplasm outside. You'll have a transmembrane protein called an SRP receptor. This entire complex is going to move over here. Your signal recognition particle will bind to the SRP receptor. So by the way, this is an SRP, signal recognition particle. That's an appropriate acronym for it. You can use that. Makes it easier. It's going to be attached to that hydrophobic portion of your protein, the signal peptide. Which is attached to the rest of the protein and the ribosome. My ribosome's looking more and more like a snowman here, but this is small. This is large. Okay. When the signal peptide and the signal recognition particle are attached to the SRP receptor, it will bring the ribosome down in contact with, no, I grabbed it, I know I did. Yes. I needed colors today, I knew I was going to. Oh, that's not going to work. Well, maybe. A transmembrane protein called a translocon. Despite my terrible drawing here, this is actually a pore. Okay, so it's a circle in your membrane. It's just a hole. It's normally closed, but when you get your ribosome in contact with this, so there's your large and your small. The translocon opens up. This SRP is going to release the signal peptide, so the SRP and the SRP receptor are no longer part of the story. This just brought this entire complex here, then releases it over to the translocon, and we're not going to talk about it again. So this SRP, this was a hot dog that we were able to use to grab the signal peptide. This will drag the whole thing over, and it will bind to an SRP receptor, which will then bring the ribosome in close enough to the translocon for it to bind. At that point, the SRP is going to release the signal peptide. So now S is no longer connected. We're going to move the translocon, and this is not going to be part of the story anymore.
So does like the SRP receptor and whole complex actually like move towards the translocon?
question is, does the SRP receptor and SRP actually move? This is a transmembrane protein, so yes, probably. All membranes are fairly fluid. In this case, we're just saying that you basically bring this in close enough to the translocon that it can grab the ribosome, and at that point, it's a signal to release.
So then would like the SRP receptor just naturally be close to the translocon?
Would the SRP receptor naturally be close enough to the translocon? I imagine so. If not, like I said, they are fluid, so they will eventually get close enough. Yes?
What happens once it reaches the translocon?
What happens when what?
Once it reaches the translocon, when does the protein then get?
So when your ribosome gets to the translocon here, it release the signal peptide from the SRP. Okay. Yes?
Once it releases the signal peptide, it goes through the translocon outside.
Yes. Well, inside. We'll talk about that next.
Does this signal peptide and the SRP detach?
So the SRP and the SRP receptor will detach. The signal peptide is still connected to the protein and will be over here. So you've got your, I'm going to draw it this way. This is your signal peptide. So when the SRP hands this whole complex off to your translocon, the signal peptide will feed through and get caught on the interior of the translocon. Okay, so hopefully you guys remember we talked about these transmembrane proteins, and we said that when you have a protein that's in the middle of a membrane, that it's going to have hydrophobic R groups in association with that. A channel would have hydrophilic on the inside to allow pass through. This is an exception. All right, this guy, because of its function, actually has hydrophobic R groups that can bind to signal peptide. You can't really see that down there, can you? Okay, so this signal peptide got handed off, it was released from the SRP, and it starts to feed through this translocon, but it gets stuck on those hydrophobic R groups, and then the rest of the protein that's continuing to be made will feed in here. Okay, so as this moves along the mRNA and makes more, you will just keep feeding it into the lumen of the endoplasmic reticulum. Eventually, when you come to a stop codon, I'll draw it again here. Nope, I need purple. And this falls apart. You release the protein will go through the translocon, still connected by the signal peptide to the inside of that translocon, and you will cleave this. So you've got another protein here that will cleave that signal peptide, and you will release this protein into the lumen. Okay, so that is going to be specifically a lysosomal or secreted. We're going to have to talk about transmembrane protein Monday. If you have a chance, watch the video this weekend. It'll help on Monday. Thanks, guys. Have a great weekend.